But with the pieces of evidence we have nowadays, we can see that there are critical inputs coming from the pain circuit, for example, entering the STN before it even reached the cortex. Then the positive thing is that I kind of rang a bell. Be careful. I can tell you that we confirm definitely on 3 macaques that we decrease their motivation for cocaine on the 3 of them. So the impulsivity is not one big thing, the control of inhibition, and STN is able to discriminate these subtypes of impulsivity. And surprisingly, indeed, the 130 Hertz did not correct this behavior, but it's the 30 Hertz stimulation that was able to correct an 8 Hertz.
Welcome to Stimulating Brains.
Hi everyone, my name is Nils Pacheco. I recently joined the producer team of Stimulating Brains. I'm so excited to work with them and keep learning. In this episode, we have a great conversation with Dr. Christelle Baunez, who is a neurobiologist at Aix-Marseille University. Christelle is known as the STN woman in France because of her expertise with this small structure. Christelle is going to guide us through this mysterious structure, the anatomy, connectivity, applications, and more. They are going to touch topics as the reward system, chronic pain, Parkinson disease, addiction, and beyond. Thank you everyone for listening, and I hope you enjoy the episode.
Okay, so Christelle, thank you so much for taking the time to do this interview with me. And I will have formally introduced you by now, so we can dive right in. I'll do that in the introduction, so we can dive right into the science. Or let's say we start not with the science, but usually I ask as first question to break the ice about your free time and hobbies. Anything you like to do when not involved in science?
Yes, I have actually, I need a second life. And that's why I'm really hoping to get retirement in our French system that is changing, because I'm a photographer.
Oh,
wow. And I like to travel a lot. So I take the opportunity at each travel, but also while walking in the streets in Marseille, I like street photography. And I organize exhibitions as much as I can. So it takes a bit of time. And I'm also interested into solidarity actions. And at the moment, I don't have much time to be involved. I've been a political activist for some years.
Yes, but, Marwan said you were a comrade.
Yes. I was actually, maybe I don't know if we should say that, but some people in the US won't even understand how strong it is, because I was in the extreme left party, the Ligue Communiste Révolutionnaire, where Marianne used to be when he was a young student in Paris.
Marwan, okay, oh, wow.
But now I've stopped that, and I will await retirement to have more time to dedicate to the others.
Great. And you have an exhibition coming up, you just said, right?
Yes,
for photography?
starting tomorrow. Every year I take part to this amazing festival in Perpignan. It's a photojournalism festival. So the official festival is called Visa, and it's for professional photojournalists. And there's an off version that is organized by the Chamber of Business and Industry. And so we exhibit in different shops all over the city during the whole festival. So it's really exciting.
Great. Where can we see your art? Is anything online? Do you have a website or something?
Sometimes I post a little bit on Instagram. I
Okay.
had a Wix web page, but I haven't worked on it. So again, as I said, I need another life or
Yes.
someone volunteering to help me.
Makes sense. Okay, great. So street art in Marseille must be amazing. I've never been really to Marseille. I think I once drove through, but I've once heard, is that still true, that there's a mural about Zinedine Zidane that was never crossed and has been there for ages, and he's a hero
It's
in the
been
city?
there, but it's no longer there.
Oh, okay.
Yeah, it
So it
was
has,
it was a black and white picture of him, not really a mural. That was on a material
okay.
on a wall. And but I live actually in the most ancient part of Marseille called Le Panier. And this area is actually the area for street art. So it's really nice because every other day there is a new thing
Yeah.
appearing on a wall. And then if you don't take the picture the first day, then it may be different the second day because some people will come and write on it or change things. So it's constant living
Very
art.
dynamic.
I like
That's
that.
cool. That's cool. So essentially the canvas is the city, and there's a lot of painting going on. Sounds great. So any other things you do in your free time? Do you have kids, if I may ask? Do you
No,
Okay.
no kids. Free time, I also like sports. So
Yeah.
and I like to hike in mountain or on the
Nice.
seaside. I mean, in Marseille, we have the Calanque, so it's an amazing place to go and hike because you have the impression to walk in mountain, but you're just above a cliff with the water of the sea at your
Nice.
feet. So it's really impressive, very nice.
Great. So diving into the professional life, who were key mentors in your career and turning points along the way?
So one of my first mentor was André Niolon, and he was a professor in neurochemistry. And actually, I discovered really neurochemistry thanks to him. And I wasn't passionate at all by that because my goal was to become an ethologist specialized in primates. And my mentors since I'm a kid were actually Jane Goodall and Diane Fossey. I haven't met them, of course, but I was reading them and dreaming that one day I will be in the field watching the monkeys. So when I arrived in Marseille for my studies, I met this professor, André Niolon, and he was a specialist on dopamine, dendritic release of dopamine he had shown with the Glowinski during his studies. And he convinced me that was really the game of asking questions in research that was the most fascinating thing to focus on, and on whatever I would work, I would enjoy it. And actually, I have to say it was true. And also thanks to people working in his lab that I really realized that. So I should mention Lydia Kerkerian-Le Goff. She has been a very much inspiring person around me. And my PhD supervisor, of course, Marianne Amalric, has been an amazing supervisor because she was really like a colleague and not like a superior person who had to teach me everything. She was really treating us as equal. And I gained a lot of confidence.
Great.
And she did a lot for me in my career. And then she introduced me to Trevor Robbins, who, of course, was a major character who played a critical role in my career because having worked with him, just you write this on your CV and you're like, wow, she's Trevor Robbins. So that probably helped me a lot to get my position back in France after my short postdoc in his lab, actually.
Okay. I think there are these websites that have statistics about, citations and so on. And he's often the number one cited person.
Which is the secret he had a secret goal that was to publish as many papers as the highest score, the best cricketer had scored, or something like that at some point. It was related with cricket record, and he wanted to have the same number of papers or things like that.
Interesting. Okay, because it's funny, I recently met him in LA and was in a cab with him and asked him that, say, I've seen this, is this true? Are you the number one cited person in the world? And he said, there are people that say this might be true, or something like that. He had a very British answer. So yeah, as if he never checks. Maybe probably he doesn't at this point anymore. But yeah, that's great. So, okay, what does dopamine do in the brain then, if you have worked with an expert?
Oops. It's complicated because but it's timely question actually, because there is a huge thing at the moment where that annoys me so much. I just posted on Twitter X my annoyance regarding this attitude that some people who have focused all their life on the role of dopamine in the reward system now suddenly discover dopamine as a key role in movement. Oh. And you're like, bloody hell. But I mean, what Parkinsonian patients are going to think about that? So we know that for such a long time.
Of course.
So I think, maybe it sounds a bit like an old person to complain about how the young people are ignorant of the literature. But please reconcile those people who have focused all their life on the VTA accumbens system, totally ignoring the basal ganglia and the nigrostriatal system, and all the others who were working on the motor system, ignoring sometimes the frontal functions and the reward circuit as well. And keep an open mind, but everything is already in the literature. It's not because now we have fantastic tools to be more selective than the main questions haven't been already asked. And if you want my point of view, nowadays science is just fashion, and it keeps coming back with the same type of questions, and except now we have technology, and technology is not everything, sadly.
I totally hear you, and I think I totally agree. I think there's this 30-year circle. I've just talked to Nico Dosenbach yesterday about the same thing happening over and over again, same questions being asked with new methods. Although what soothes me is, so Haggai Beckman has a really good book about the basal ganglia. I like it. It's a great primer, and I think he even really confronts the movement basal ganglia diagrams with the reinforcement ones and tries to marry them somehow, and kind of succeeds. Also with good jokes and in there, as you can imagine. And
but Wolfram did in a way. Wolfram Schultz, when he published his first papers on dopamine and reward prediction error, there was no dissociation between VTA and substantia nigra compacta. His dopamine neurons were from every dopamine nucleus, so with no distinction.
I totally agree. And no, I wanted to actually say, so in his book, he discovered that, things he discovered were known since 30 years, and he didn't know that, right? So even people like him suffer this problem that 30 years ago, there was somebody already, right? So it's always easy to look at the young people and say they don't know what they're doing. They haven't been around. And of course they haven't, right? It's very hard for us. I'm not that young anymore, but, I used to be in. It's very hard to know the old literature. It's so easy, right? You need a good mentor for that. Now, if you don't, you essentially think, oh, this might be new, right? And very, very not even so easy, even with PubMed, to go back and find exactly the paper that did this really well in the 30 years ago or 20 years ago. So I'm just suspecting the same might have happened to you guys when you were young too. So, because there was for example, there was so
I had excellent mentors.
Okay.
Especially the one I cited from my PhD time. Every original paper had to be mentioned in my PhD thesis. I couldn't just rely on reviews that would make the summary of everything and just quote reviews. It was forbidden. I had to go and find the historical papers and give credit to those who were initially at the origin.
That's
So
fair.
it was
And
really
I think
tough with me, but that's why it's a really sensitive point to me.
No, it makes sense. And you're probably very right that, at the time, science was also a bit slower
But there
in a way,
was
but more
less
thorough.
literature. It
Yeah.
was easier to follow and to go back. But
Yeah.
now, if you type dopamine, reward, and movement, as now they rediscovered that, if you type this, you're going to have 1,000 of pages on PubMed.
Yeah.
And
Yeah,
the
you're
young
right.
don't start with the first page. They usually start from the last publication.
You're
So for
totally right.
23, you're going to have 300 papers, and they can't. It's our responsibility because we let the system go this way, but too many publications kill the knowledge.
You're totally right. It's a problem. I really see that too. It's hard to keep up these days, even with the new stuff. And then anyways, let's go back to science and your expertise. I think so. First of all, I want to acknowledge my postdoctoral student Garance Meyer from France. She knows you. She helped me a lot with preparation of these questions. So I want to give her credit here. And she kind of said informally, you're known as the SDN woman in France and internationally. So I think you really like that structure, the subthalamic nucleus. What does it do in the brain?
That's a tricky question. First of all, yes, I want to say I really identify myself as a STN obsessed person. And to such a point that when I hear someone mentioning STN, I have the impression they're talking about me. So it's a disease. A psychiatrist listening to us, be in touch with me. I'm your subject. So what does it do? It's complicated because for many years, when I was introduced to this little nucleus, it was considered as a simple relay on the indirect pathway of the basal ganglia, playing mostly a motor role, since all we knew about it was that when there was an infarct, a spontaneous infarct within the STN, it would induce these ballistic movements in patients. So that was obviously a motor related structure. And then progressively, when it started to be in the game of Parkinson's disease treatment, we suddenly looked at the consequences of manipulating this nucleus. And that's when I entered the game. In 93, 94, I started to make my first lesions in the rat. And I could see they did not only have motor problems, there was something else. And then I read the anatomy. And looking at the connectivity of it, of course, it made sense that we were always taught, the students of the basal ganglia, we were told about the parallel loops, but we were focusing on the motor loop and totally neglecting the other ones. And then actually the STN was not even drawn on the parallel loops.
Yeah.
But it is totally connected with directly with the prefrontal cortex, with the ventral pallidum. So it had to play a role in frontal functions. And that's when I decided to investigate that. And then I discovered that as small as it is, it is involved in many, many functions. Of course, you may say, I mean, most people are cortical driven. So they will always put the origin starting point at the cortex level, and then all the subcortical structure are only simply relaying information. But with the pieces of evidence we have nowadays, we can see that there are critical inputs coming from the pain circuit, for example, entering the STN before it even reached the cortex. So you
from below. Oh, I didn't know that.
Yes, from below. So from the cerebellum, from the parabrachial nucleus. So there is a nociceptive network that has been highlighted by Véronique Coiset in Grenoble, who is actually dissecting, and I contribute as much as I can to understand how. The question initially was why Parkinsonian patients suffer from chronic pain. Not all of them, but many of them.
Yeah.
And it's been ignored for many, many years because we didn't have a clue of what was going on. And in fact, she discovered actually with Peter Redgrave and Paul Overton that there was actually a pathway involving the STN. So that may be a way to enter the basal ganglia with nociceptive inputs. And then, of course, there are connections with the amygdala, so giving emotional information. And then there are some recordings showing such a short latency that it cannot possibly just go through the cortex all the time. It has to be really direct inputs coming to STN and where it can filter. So I think I'm never really good at addressing the question, what does it do? It does a lot because it's in a position of a nexus in the brain, receiving so many information and also controlling many things. But it has possibly, because we noticed in recent papers where we did some recording of STN neurons, that these neurons are the only one in the monkey we could find. We compared different subpopulations in the striatum, codeputamen, STN, globus pallidus. Only in the STN could we find neurons that could both integrate information regarding the value and the cost. So this integration was found at the level of neurons in the STN. So it's kind of an integrator, and it can possibly be serve as a facilitator. So like a gate control, I would say, on many processes that are still not yet all investigated. So our recent one is on social information and social.
We'll get to that. So this is a great. So it makes my head spin, right? Because I always tell my students, STN receives input from the entire frontal cortex, which already is a lot, right? Not just from motor and so on. And a bit like the thalamus in small, right? It's really, it gets information from everywhere. Which, and it's not so big. So you tell me about all these other things with pain and social things. It's too much for this small structure, right? So what does it even do with all that information? And you said something like integrating nexus and the facilitator, which goes in line with what I've heard from other experts. So Haggai Beckman once said something that I really liked, and maybe you agree or disagree, that he said, if you think of a piano, the STN is the pedal to facilitate, but the striatum is the melody. I don't know if you like that, or because it would be similar to essentially loosening the brake, right?
I remember listening to one of Haggai's talk, and he was using this image. I kind of like it, except the only reserve I would have with this analogy is that some people play piano without using the pedals. And I think that if you want to live with no STN, you're a kind of person highly disinhibited, probably, and with many possible problems.
That makes sense. So, and you need it, right? I mean, well, anyways, so I think going into that, you, after the PhD with Marianne Almeric in Marseille, you did a postdoc with Trevor Robbins in Cambridge, as we said, and then I think in that time you found out that while lesioning the STN reduced reaction times, it also played a role in response inhibition and reward processing, as you've just said. So I think you really put the non-motor STN on the map for many people at that time. But I think it was, and also lesions caused attention deficits, impulsivity, perseverance, as you've just alluded to. And this was, I think, around the same time that STN DBS was started in Grenoble.
Exactly.
For Parkinson's. Were you worried?
Actually, yeah.
Were you worried about that at the time?
I was not worried. I was concerned by the fact, first of all, my first paper on STN came out in 95, exactly the same year Patricia Limousin published the Lancet paper with the DBS applied in PD patients. And we had a huge community in France of the basal ganglia research, so I knew already about that, and they knew my results in the rats, and I have to say that Alim Louis Benabid didn't like my results at all, because I was saying, okay, inactivating STN is good for Parkinsonian akinesia, because our rat model was a reaction time measure. So that was really good in alleviating the akinetic problems. But then we induced something else. And so they are not normal. And I suspected it would have something to do with cognition. And that's why I wanted to go and work with Trevor to really develop cognitive task to assess the role of STN in these functions. And Benabid wanted me to be outside the STN because he didn't want me to find out there could be possible side effects we didn't want to see in the PD patients. So that was I was not worried, but I was concerned that they didn't really want to see side effects. Maybe you should cut that.
Well, no, but
and they won't like that part of history. But anyway, then the positive thing is that I kind of rang a bell. Be careful, because in the patients, you may recover some of the dysfunctions the Parkinsonian had, but you may induce something different, or at least check how maybe they can be improved on some other aspects, but STN is not only the motor structure you think it is. And then progressively more and more neurologists were telling me when I was in meetings, oh, by the way, I have a patient is doing this and this, and there were all these stories of incredible side effects they could see after manipulating the STN of patients. So that was really kind of satisfying to see that even though my work was on rats, some clinicians were paying attention to what I was saying. It's not the case for the majority, I should say, but some were really interested because I was really digging into what do we do when we inactivate STN. And I had always that in the back of my mind. It was always to better understand what could be the possible consequences in the patients if we manipulate their STN.
So you predicted it, it came true. Did Paul Crack play a role in that? Because he's now quite, that's his field. It has become his field too.
Paul never liked, he never liked to attribute the side effects to the STN. So even for the motivation and the apathy effect he has, he doesn't want to accept the fact that STN has something to play in it. He says, oh, it's just because we reduce the dopamine medication and then now they are apathetic. But it's a Grenoble school. So,
interesting. I really always thought him to be more, that's what he focuses on the non-motor effects of STN, DBS and so on. But I'm might have also changed over time, but
no, he, of course he's interested in that, and we exchange a lot, and he's a good friend. I really like Paul a lot. But on the discussion of the apathetic effects described in some patients, I was always telling him, no, but it's normal, because STN is involved in reward-related processes in motivation. So it's not a surprise that the patient should change their level of motivation. Into which direction is a big issue, because it might be dependent on what they were before the surgery.
So you alluded to it before in the classical reward circuit that we know is published, for instance, by Wolf 2002, Morganson 1980. You had that in your slides when I saw you talk in Grenoble. The STN doesn't play a role.
No.
Do you agree?
Totally ignored. It's been ignored all the time, and even in the recent review by Christian Lucher, you mentioned that in the email we had, and Christian knows very well what
but-
we are doing, and he
but
is
let's-
also working on it.
We'll go into addiction next.
Okay,
I mean, maybe for you it's the same thing, but let's start
But
with.
no, it's not the same thing. But all the people working on addiction work on a classical view of the reward circuit in the brain, and all the academic books you will open describing the reward system will never mention the STN.
Yes.
At the Mogensen time, but also reshaped by Marina Wolf in 2002, there's still a box with motor outputs and accumbens position as the interface from motivation to action. But if you look at the connectivity we now know, because we look more carefully at the STN, we can almost make the parallel between accumbens and STN. And I also always play the game on my slides to add the STN in this diagram and to show, look, there is input from the prefrontal cortex, there are inputs from the dopaminergic neurons, there are now inputs from BLA. So the only ones that are not described are from the hippocampus. So learning memory is still something that is not that much affected, but otherwise everything else is when we touch STN.
It should
be, but
it should be, and I think, you know, learning from the motor system, we know how dramatic the effect is, right? So that's for neuromodulation. It's a small structure, but for neuromodulation, the STN is an amazing target. Because it's small, because our electrodes are small and so on. So it's a critical thing. And if it's really the facilitator for lots of things, that's exactly what we want in all of these systems in a way. Or we want to be able to up and down regulate that, I guess. So maybe speaking about that, why do you think the small structure has such a dramatic effect? Does it have to do with the funneling effect of compressing the information and then there you reach a large network? Or is it more just the way the STN is wired? Because I think if we would put an electrode into the striatum, we'd also see effects, but it's likely more specific, less global, maybe?
Yeah.
Right? Or what do you think about this?
There are different aspects in the answer. The direct connectivity surely plays a role. This description of functional territories has been done. I'm not an advocate of the contrary. I think dominance in terms of functional territories exists, but at least in the rat, the way the neurons are so compact, so massive dendritic arborization covering the entire structure. So you lose a little bit of specificity. That may be why wherever your electrode is going to be, you will recruit neurons involved in a network you wouldn't want to touch.
Yeah.
So that's the difficulty. In human, apparently the target is different for Parkinsonism than for OCD, and manages to minimize a little bit side effects you wouldn't want to see in PD patients or the other way around in OCD patients. So it's possible that there is a selectivity, however, but it's small, then compact, and therefore synthesizes many functions. Now, the other thing I wanted to say was the passing fibers going through the STN. And I'm not an anatomist, but I'm a big fan of Susan Haber's work. And she's described for many years, be careful, because depending on where your electrode is going to be, you may stimulate fibers going to places you would have no idea they could be related to STN manipulation. So this is really critical, and it's really surrounded by fibers. So it's not only the direct connectivity of it, but it's also the location of it in the brain that makes it really susceptible to affect many behaviors.
Great. I mean, I sometimes think because of that, you could conclude from that it is a bit of a dirty structure, right? Because with for neuromodulation, because there's kind of everything there. So and we have the side effects, but they are not that common anymore, at least, or like it depends on how subtle they are and so on. I sometimes think, why is that, right? Why is STN-DBS so forgiving, and why does it work so often? Because the surgeons don't always target exactly the same millimeters. That's my field. I sometimes think it has to be because it selectively targets broken circuits. So things, for example, where there's a feedback beta signal that is most affected by STN. So even if you're slightly off that circuit, you would still have the most effect on that symptom, because that's the quote unquote broken one. Does that match your experience, or is it really about just where you are and that's the symptom? Because maybe to add one more data point, if we put an STN electrode into OCD patients, they don't get hyperkinetic usually, right?
No, it's true. But hyperkinesia anyway is always transient. So when you induce it, that's the way we use the checking of our location of electrodes in the rats, because in the rat it's really a tiny structure. So after 25 years working on this structure, we still struggle, and sometimes we get only 60% correct surgery in most of our experiments. So one way to check it is to induce hyperkinetic movement, because we work on intact animals most of the time, and that's a big dissociation between when we're talking about PD patients and my data, because I work mostly on intact animals, and then I don't repair a broken circuit.
So do you induce hyperkinetic?
Yes,
with
we
130
induce.
hertz?
As I said, the historical work published in the 49 by Mettler was describing an hemorrhage in the STN inducing hyperkinetic movement. So when you induce a selective inhibition in the STN, you see the rat making an ballistic movement of the contralateral paw.
Yeah.
This is well known, and it's a good way to say, okay, our electrode is well located, perfect. Which, if you go in any other structure, you don't have such a fantastic control. So it's really good, but it doesn't last.
So you're totally right about ballism. That matches my experience. But like these hyperkinesias or dyskinesias in Parkinsonian patients, right? That if you have too much levodopa and STN stimulation, people are dyskinetic, and that lasts usually. Now, if you tune down the voltage a little bit or reduce the levodopa, it gets better. But I think we never see that in OCD. At least, I mean, I'm no expert there, but right. So where
it's more sensitive to levodopa, isn't it? I would have thought so, that GPI was more critical for dyskinesia induced by levodopa than STN targets, but I may get this wrong picture. But no, to go back to the initial question, really is how, with such a general intervention that is not that specific, we could possibly repair without affecting further. Is a big it's a big issue we are not able to address nowadays. And I think as many Parkinsonian subject types there are, as many effects and side effects you can see with STN-DBS. That's the difficulty, because they all have their own history of treatment. They all have their specific type of response to levodopa, and they have other treatments. They experience many different things in terms of social life, etc. And now we see in our rats that playing with STN can also affect the way you see others. So you see so many consequences you could observe in some patients, but not in some others. And we are not able yet to really assess and understand why it could be different.
A guest question by your comrade, Marwan Harris. Given your several nice papers describing the role of the STN in various emotions, where would you place the SDN in the grand circuitry of emotions described by Papez in 1937, who does not mention the SDN in his emotion circuitry? Similar question to before, but
yes,
it's a different literature.
different
So
circuit, but it's the same. Yes, although while I'm really strongly advocating to introduce STN to the reward circuit because it really deserves to be there, because we have shown really, and others now replicate some of our findings. It's really critical for these functions. On the side of the Papez circuit and emotion, it's more discrete, I should say. And I was referring to the BLA projection to STN. It's been described in one or two human brain imaging paper as a functional connectivity that was modulated in some tasks. But when we did some tracing in rats, it's really kind of anecdotal. So doesn't mean that it doesn't play a role. But it's not as strong as the involvement, I think, of the STN in the reward circuit. So it should definitely be in the Papez circuit as a structure in interaction with the BLA and central amygdala as well. So amygdala in general. As I said, hippocampus, not much as far as I'm aware.
But Papez circuit, I think, was originally coined as an emotional circuit, but we think of it more as a memory one right now, right? Or so is that still correct? So I think it's probably not so 100% black and white, but yeah, okay. So that makes sense. So now second guest question by Marwan is, do you know who said that the basal ganglia are the site of motion and emotion? And then he adds in brackets, motion and emotion has been adopted by the car maker Peugeot to advertise their cars.
Yeah, I don't know. I mean, if Mogensen was considering the accumbens as part of the basal ganglia, he was certainly the one talking about an interface between motivation to action. So that could be my answer. If it has to be more recent in the fashion, as I was saying earlier on, people are rediscovering.
Yeah, no, it's not that recent. I mean, he revealed the answer. Then. I have the answer now. Yeah, it's apparently Eva Ajit, according to Marwan.
Eva Ajit, okay, yeah.
And maybe Haggai Beckman, he said. But Haggai
I was
CC,
tempted to say Haggai could have said that, but Eve, yes.
Okay.
So I don't know if all if the people who are going to listen to this know who Yves Agid is, but he's one of the most critical neurologists in France in the 80s, 90s, who contributed so much to the interface because he was a neurologist at the Salpêtrière in Paris. And he trained so many neuroscientists as well. So he was really one of the key medical doctors being at the interface with fundamental research, very interested in research and bringing the clinics to us, neuroscientists. Yeah. So he's been a prominent figure.
Yeah, he's a legend. So yeah, I think totally people should know him. I even tried a lot to get him on the show, and try to reach him. It was Marwan's suggestion, but so far I haven't reached him. I think I have the email address, but didn't get a reply. So yeah, he's again, as you said, very important figure. Yeah. Very knowledgeable.
He's written books. He's been really important at creating this huge institute in Paris, Salpêtrière, l'Institut du Cerveau et de la Moelle. And they were actually pioneer at bringing Mécène into science in France. So their first sponsor were Jean Todt and Michael Schumacher before his accident, actually.
Oh, wow.
Sarcastically. So they were the major sponsor, and Yves has played an immense role in the creation of this institute.
Speaking about frontal cortex, in the Baunez 2011 paper, you wrote that the SDN has frontal cortex-like functions. Again, same topic, but we kind of often see this inhibition. Not often, fortunately, but we sometimes see it with SDN-DBS.
Yeah, it's very interesting because when you inhibit STN into whatever you use, lesion, electric DBS, or optogenetics inhibition, we find a lack of inhibition in terms of impulsivity of action. So the animals or subjects have difficulties at holding a response. They can't wait. They have to move. But the contrast is if you give them a choice and then you measure impulsivity of choice, then you get the opposite. They become suddenly more conservative in task. So now maybe I should be more precise. Typically, when we talk about impulsivity of choice, we refer to tasks like gambling. So the Iowa gambling task, where you have to choose options, and optimally you try to get the larger reward with the best probability. And or delay discounting. In the delay discounting, it's a task, you may have seen the video of the little kids with the marshmallow.
Yes.
So if you wait, and if you want to eat now, you have one marshmallow, but if you manage to wait, you will get a second one, and you see how they have to cope with the time lapsing. Yeah. So when we do that and we inactivate STN, actually they can wait better than controls.
Interesting.
So because they're so highly motivated for the reward, I think that may also play a role in that. So the impulsivity is not one big thing, the control of inhibition, and STN is able to discriminate these subtypes of impulsivity. So you really move a brake on impulsivity of action, and they can never wait. Stop task is a nightmare. They can't stop if you remove the STN. And actually, in functional imaging studies, it's been shown that the STN lights up when you perform a successful stop. And we have recent data that we're going to submit in the next weeks on functional imaging in drug addicts doing a stop task, and we focused on the STN, and it's really brilliant because it really works. So it's really involved in that type of control. So you remove it, you lose it. But when there's a choice involving reward at the end or avoiding a punishment, which is a kind of reward in a way, then they will be able suddenly to control their movement and weight. So that's
very interesting. So one is
discrimination.
They're different time scales as well, right? One is more about direct action. The other one is more about delay discounting. And you say the important thing is to have a choice. Yeah. Okay.
Introducing a choice makes a difference because then you have different outcomes, and the STN is involved in the valuation of cost benefits.
That makes sense.
Yeah, you are. It's worth waiting a little bit to get a larger reward, so they do.
Interesting. That's very, very, very insightful. So I always remember Peter Brown from Oxford. You probably know him. He always said there was one introduction sentence. He said that if a cat lures for prey, right, they have to be very stiff. They have to be, essentially the engine's already running, but the brake is full on. Apparently beta power is very high in that state in the STN. The stiff Parkinsonian but physiological state. And then if they run for the prey, then the beta drops.
In this case, it's interesting because it's exactly, I was thinking while I was getting excited to tell you all about this control of inhibition and choice, that in activating the STN, then if the STN is valuing, then it should be disturbing, and their choice shouldn't be optimal if you destroy the STN. But I think that's where all the work by Peter is really critical, because it's not always a matter of total inhibition or activation of something. It's modulating subabnormal activities that can be found in STN. And when we were talking earlier on of repairing a system, actually in PD cases, you have this abnormal beta oscillatory activity in STN, and applying DBS, we've shown that in our rats in the context of addiction, but it's the same. Peter has shown that as well, that you reduce this abnormal activity when you apply DBS. And that's one explanation why it works.
Yeah.
You diminish an abnormal activity.
Can we simplify it even further and say this high beta powers overactivation? Probably not, right? We shouldn't simplify that much.
No, not necessarily. Yeah.
Yeah. Okay. I'm a lumper. Sometimes I like to be and try to make things as simple as possible, but sometimes, of course, yeah, that's not great. All right. So for a long while, I think your main contribution, and we talked about this, has been that to show that the SDN has non-motor functions and should be considered part of the reward system. Now, think a bit later, or maybe around the same time, you also thought about addiction, and you've alluded to it before. I interviewed Christian Lüscher in episode 20 of this podcast, and he's the author of what is often called the standard model of addiction in the brain. Again, in his wiring diagram, the SDN is not in there, not even in the revised version of 2021. I ask again, do you agree? And we already know you don't, but
no,
what is your what is your work about addiction?
And I really don't understand why Christian didn't add it, especially in the revision of it, because I mean, he knows perfectly well, and he's working on it nowadays. So he should really.
He's treating SDN-DBS patients in Parkinson's himself. He's you know that, right? He's a neurologist.
Yes, yeah. I mean, in PD patients, yes, but also in the context of addiction, he's done some work recently on the STN. So it's a bit weird to continue to ignore it if you suddenly are interested in working on it. So, but I have no doubt in the next edition he will introduce it. Maybe
we'll be there. Okay.
Or he could be. At least they could mention it as a provocative thing, saying things don't stay always like that. And now we suggest that it could be implemented with STN on the diagram. But yeah, it's actually while I was when I started to observe my rats, and I noticed they had some strange behavior towards their food reward when I was feeding them every night in Cambridge after my experiment. I thought they have something strange with the reward, and they were always checking in the magazine in the tasks they were doing, the attentional tasks, the 5-choice tasks.
These were lesioned STN rats.
They were lesioned STN rats, and they were always checking if they had rewards. While they knew they hadn't performed well, but they were keep focusing on the magazine to go and check. So I thought there's something going on with motivation, and I need to investigate that. And I was particularly, again, concerned thinking always at the Parkinsonian patients. And then I thought, if we modulate the reward system in these patients while they're taking dopamine medication, we may induce dramatic effects, because what I was seeing was an increased motivation for food. Mm-hmm. So I thought, and again, I was referring to my textbooks, the reward system is unique. It's all about dopamine, VTA, accumbens, blah, 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 and it works the same way, whatever the reward on board. So if I see my rats having an increased motivation for food when I inhibit their STN, if we kind of inhibit the STN of patients taking dopaminergic drugs, are they going to be much more dependent on them? Former alcoholics, are they going to go back in precipitating their alcoholism once they receive the STN-DBS? So that's the type of question I ask. And that's how I entered in the field of addiction with my own research, because I thought I really need help of someone doing self-administration in rats to check if my lesion is going to affect the drug intake in rats. And that's when I started to collaborate with Martine Cador in Bordeaux, and that I published this really puzzling result, because actually I expected the total opposite. And then so I told Martine, be careful, because I'm coming to do the lesion in your lab, but I won't stay for all the experiments. So if when I leave, be careful every day, because the rats may overdose. They may go crazy for the cocaine, so we have to be careful that they don't kill themselves. She said, okay, fine. And then I was receiving messages from the students. There's no difference. They don't show any increase in their drug consumption. I said, okay, fine, perfect. It's really good, because whatever would have been the result, it was interesting anyway.
Yeah.
So interesting for the clinic if it had no effect. Interesting for our knowledge if it was increasing. And after, so I was telling her, we have to wait for the histology, because your results show a big variability between all individuals. So let's see after histology. So she sent me the slides. So I had no clue who was taking a drug or not. And I checked the slides and I said, this one is not well lesion, this one has a good lesion, this one has a good lesion, etc. And then at the end, she reanalyzes the results and she sends me a message to say, by the way, your hypothesis was wrong. We have the opposite. Now they decrease their motivation for cocaine when they have an STN correct lesion. And I was like, wow, that's fascinating, because nowhere else in the brain you can induce this opposite effect, increasing motivation for drugs, for food, and decreasing motivation for drugs. And I was like, wow, how am I going to explain that?
It's a perfect time for guest question by Haggai Beckman. He says, hi, Christelle, I look forward to your stimulating talk on the Stimulating Brain series. I'm sure you will touch on the issue that SDN-DBS can ameliorate the desire for substance of abuse, but not for natural rewards like food and sex, which you just said. Do you understand the neuronal mechanism of this dichotomy? Are there different neuronal circuits for the substance of abuse and natural rewards, or is it the same circuit but with a different intensity of coding? And I think that was your Nature Neuroscience paper 2005.
Yes. Yeah, that's exactly this paper. And thanks, Haggai, for asking this question. It's a tricky one. I have to confess that it's been occupying my my mind since all these years, and we have partly some answers. So what I should say is that STN increases lesions or inhibition of STN increases motivation for what is beneficial in terms of calorie. And we've noticed that they make difference when they work for sucrose that is caloric and with a sweet taste. But then we did experiments with saccharin, where you have the calorie but not the taste, and glucose. Saccharin, you have the taste, not the calorie, and glucose, where you have the calories but not the taste. Rats love the sweet taste immediately, spontaneously. But when they get to understand that this sweet thing doesn't provide any calorie, they can shift their interest. But you need to make them understand. And when you give them the choice between saccharin and glucose, we actually showed that STN lesion rats shift more quickly than controls to the glucose solution, because they understand the caloric intake. And as soon as they understand it, they shift more quickly. So they don't stick to the sweet taste of saccharin. They don't have the same, maybe they don't have the same pleasure at leaking saccharin than controls. But anyway, we found that maybe the explanation of this increased motivation for sweet food was that because we were using sucrose and it is caloric. And it's been shown by a neurologist in Clermont-Ferrand, Frank Durif team, that Parkinsonian patients with ST and DBS have a change in their metabolism. So the interest for calorie might be something that we tackle when we manipulate STN. So that could explain some of the difference. However, we have also shown naively, when I first had these opposite effects on motivation, I thought, oh, maybe there are different types of neurons in STN specialized at encoding a specific type of reward and another type, and then they don't inhibit the same way. And that could explain why we have motivation in one direction, motivation in the other.
Mm-hmm.
So we did experiments, type Wolfram-Schultz type experiment in rats, where the rats were trained to press a lever, get signal indicating you're going to get sucrose, you're going to get cocaine. And then even before the reward was delivered, we could see activation in STN that was changed depending on what reward was announced. And it turned out that nearly 50% of the neurons we recorded were responsive only to the cue predicting cocaine, and the other 50 were responding to the cue predicting sucrose.
And no overlap? Were they always selective, or were there
most
some?
of them were selective.
Okay, interesting.
Only, so I said 50-50, it was 45-45, and that it was approximately 10% of neurons that were responding to both cues.
Okay.
The thing is, in STN, nothing is simple. So the specialized for cocaine were not going in one direction and the sweet neurons in the other direction. They were all responding with inhibition activation at the cue presentation. So it's not as clear-cut as the dopamine responses or any other neurons in other structures. The STN, you always find all type of responses. So it couldn't really address the question whether or not there was a sub-circuit that could mediate the drug-related reward and another circuit for the sucrose and the natural reward type circuit, which was also something I think Tricia Janak may have worked on for some time. And no, there was another work anyway, describing in the accumbens different population encoding different types of reward. But I mean, I'm not really sure. It does have to go through different circuits, but it's simply maybe encoded in a different way, as we know now that even depending on the type of drug, you may have different behavior if it's opiates or psychostimulants. And it's not only a matter of specificity of the neurochemistry. It's, I mean, the reward circuit modulates differently.
So, but on a behavioral level, I think in your 2010 paper, PNAS paper, you doubled down on this idea that like lesioning or ST-NDBS in rats, I think at that point decreased the motivation for cocaine but increased the motivation for food. That was replicated in monkeys 2016, and I think that ties into the work you
the replication in monkeys is not published.
I'm sorry about that. So I was given.
So we are actually writing up the paper.
Wrong information. Yeah,
but
okay.
no, we have had abstracts on that experiment in the past, but no one else, as far as I know, has looked at that in monkeys, and we are. I can tell you that we confirm definitely on 3 macaques that we decreased their motivation for cocaine on the 3 of them. The increased motivation for applesauce was more difficult because they were not at equivalent level of motivation initially for this reward. But the trend is actually quite encouraging, and we can say we sort of replicate.
And it ties into what you also did with OSCBO, I don't know if you were part of it, but you have it in your talks always, that that weight gain of ST-NDBS in Parkinsonians. So maybe you can is there a way to summarize that together, how you think about increased appetite, increased and then decreased cocaine abuse?
Definitely, yes. I mean, it's been observed in some patients gain weight. That's why we think it may have to do with a change in the way, in a metabolic change that makes people seek for highly caloric food supply, and they go for chocolate a lot and sweets. Those patients with this obsession with food. So yeah, there is this effect that has been found in the clinic. And on the drug issue, we have a few observations of Parkinsonian patients suffering from the dopamine dysregulation syndrome. So they are diagnosed equivalent as addicted patients to their levodopa treatment. And we found a beneficial effect on that DBS of ST and on this side effect of the levodopa treatment, actually. But I was talking recently with Alexandre Zebio, who published this study on the DDES patients, and I was asking him, so how is it going on? Because nowadays, the posology is changing. People are more careful about all these DDS. And he said, yeah, in fact, we don't have that many patients now because
people know it's posterior. Yeah.
The clinicians are more aware, so they pay attention, and then so there are only few patients still with DDS issues, but less and less. So we won't have. We can't rely now on these Parkinsonian patients to have observation on addiction-like behavior, but it's about time to try on real addicts. The difficulty is to recruit them.
Yeah, we get into that in a second. So maybe one last thing. I recently had lunch with Paul Sisek in Montreal, who is currently writing a book. I think you know him. He's also runs a monkey lab in macaque lab in Montreal, and he mentioned that one chapter of his book will be about the orbitofrontal cortex, and he's always looking back in time with his phylogenetic refinement approach, so evolutionary time. And he said that the OFC, we often attribute the function of encoding value to it, but looking back through evolution, it was more originally had the function of encoding food and maybe calories. I was thinking of that while we're talking. And so it's also, I think, just next to gustatory cortex and contains secondary taste and olfactory cortices. So maybe during evolution, this function that was originally just food encoding, like food value encoding, was extended to more abstract encodings of situations, valence, and is this a good thing? Is that or is it a good situation, bad situation? So, and we know that there's a hyperthalamic pathway from OFC and ventromedial PFC projecting directly to the STN. So, speaking about the circuits that Haggai asked, could there be a slight distinction of specifically the input of the STN, one more encoding for calories, the original, let's say, OFC function according to CSEC, and one bit more whatever dorsal that might be encoding for other types of valence? Do you see a...
Yeah, I remember Léon Tremblay with Wolfram Schulz that published nice work on how OFC neurons were encoding the relative preference of different juices for macaques. And yes, of course, I was thinking at all this work when I did my recording in STN neurons, trying to see whether there was a preference and all these valuation possibilities when we implemented the task for the monkey with the force to apply and see the modulation of all this and the timing of our responses. To me, is so short that as I said earlier on, I'm not sure it needs to go through the OFC, but the connection, although it's really little in the rat OFC STN, in the primates, it's well represented, and in the human brain as well. And interestingly enough, they really have a lot in common, OFC and STN. And in a review we had written in 2010 with Donegal, we had reviewed all the work carried out with lesion pharmacology using the same tasks in rats. So that was a rat review on control of inhibition. 5-choice serial reaction time task, so with premature responses as impulsivity of action, perseveration, delay discounting, stop task, all the tasks reflecting impulsivity of action and the one impulsivity of choice. And what make each lesion, what are the results? And we compared. And of course, we were in Trevor's lab at the time, so he was obsessed with accumbens in control of inhibition, and we could not find the evidence in the literature, even in his lab, of consistent lesioning of the accumbens disturbing this behavior. In contrast, OFC, dorsal medial striatum, and STN were the three structures constantly mimicking exactly the same deficit in the same task. Regarding these aspects of behavior, and not exactly the same deficits as a whole task, but specifically on the premature responses, any measure that are typical of control of inhibition. So the OFC, dorsal medial striatum, and STN is a network that is really critical in most of the behavior we are interested in. Control of inhibition, decision-making by extension, and valuation of reward. Why not? Because that's exactly what they're doing.
Interesting. Dorsal medial striatum. Okay, yeah, it matches so from our results, we have a I looked at just where we need to stimulate for, let's say, OCD responses in humans, and STN works great, sweet spot there, perfect site, but then the NAC does not. So the accumbens is not a good target. And I think, the electrodes are often, with the tips in the nucleus accumbens, or sometimes even more posterior and the ventral pallidum, but the actual, contacts that people look at are more in the anterior limb of the internal capsule, yes, which projects to the STN, right? So it's more like, seems like many of the, ventral striatum targets there seem to be in that same circuit you just described, but rather stimulated the white matter in the capsule that would project to the STN from these sides. So it was just I have to dig up that literature. That's really, really interesting, I think, for what we do too.
But we said right from the beginning, I'm obsessed with STN, but of course I don't naively think that he does it all by himself.
Of course.
And its connection with the prefrontal cortex is really critical. So that's why now, with the fancy tools everyone uses, and I had pressure from editors to stop the lesions and move to optogenetics and fiber photometry. So we have now these tools in my lab, and we're looking at the specific contribution of the particular subtypes of hyperdirect pathway and what is their specific contribution to the different behavior we're studying. So anterior cingulate cortex STN, prelimbic STN, infralimbic STN, etc. So
great. I wanted to ask that more generally. Do you work mainly with rodents now, or also primates?
I do primate a little bit. I mean,
okay.
In my team, Paul Apicella is still working there on his TANS neurons. So we still have some macaque activity, and I have project to use marmosets.
Okay.
but it will be for more social context approaches on the addiction field. I mean, everyone is aware that there is a big difficulty at keep working on primates nowadays, and especially at acquiring macaques is a big issue. So, and it's also, as I told you earlier on, my passion is monkeys, but in the wild, and I wanted to be an ethologist. So in the lab, I have to confess that it's been really difficult for me when I did myself experiments with macaques on this deep brain stimulation experiment. And they had also self-administration of cocaine. So it was a heavy experiment. It was difficult. So as long as we can do really funny things, and I have fun really doing my experiments with rats, and we have a translational value in our work, and I still want to believe that. So if I can minimize my impact on primate research, personally, I do. I of course will always support the people doing it, because I think it's useful. And I have now the position of chair of the care committee of FENS. So that the care committee is the committee on animals in research. And then I have to actually, I went to the European Parliament to talk with the politicians to explain them why we still need to do research on animals. And it's really important. And it's also very important to support research on non-human primates, because there is no way to replace at the moment, and especially for brain research, what we like. So, but if I can avoid to do it myself, it's better. But I really support totally what Agai is doing, for example.
Totally agree. No, I mean, it of course has to be, as minimal as possible, but I think you can't replace it. I totally agree with you. So, and the for the optogenetic stuff, I think you would use mice though, right? Not rats.
No. We do everything with rats. Okay. So I have to say that I'm a rodent person, but a rat person. I don't like mice, because they're really difficult to train in behavioral tasks. They are thick, so you have to change all the parameters. So an attentional task to me is no longer attentional when you have to increase too much the stimulus duration for a mouse to be able to perform, etc. So, and they stink and they're small, and no, I don't like
they
that.
Stink. Love it. Okay, that's like it. Circling back to addiction for a second. Does stimulation frequency play a role? I think you investigated 130, 80, 30 and 8 hertz stims. What are the effects of each of those in STN?
Okay, so yes, it's very important point, because it was kind of a surprise to me that 130 hertz used to work for many aspects of what we looked at. First of all, when we started to use it in the context of Parkinson's disease, of course, when we developed the first behaving rat subjected to DBS, we applied, of course, 130 hertz. And then we tried low frequency. We didn't have a beneficial effect, but we didn't really see detrimental effects. It was not an opposite clear-cut effect, at least on the task we were using. After, other people have published, studied, really assessing the parameters, modified, etc. So that was a common sense to consider that low frequency would stimulate the neurons, high frequency was inhibiting the neurons while stimulating all the network of fibers passing through. Okay, I'm fine with that. Still, some people want to debate on that mechanism. To me, it's boring. So I accept this general concept as, functionally speaking, in terms of behaviors. That's what I see. The outcome really looks like an inhibition of the structure in many behaviors, not all of them. Anyway, so I thought when we are going to do our treatment for drug addiction, 130 hertz should be efficient for all aspects. And it's not actually what we see exactly. So to give you a really brief summary, 130 Hertz works perfectly fine on the measure of increased motivation we were referring to before, with the monkey data and the rat data. It also works at preventing the loss of control over the drug intake in a model of escalation of cocaine intake. It works with cocaine, heroin, alcohol, on this measure of escalation that is prevented. And once the animals have escalated, we can correct by decreasing their level of drug intake, again, stimulating at 130 Hertz. So far, so good. But then when we move to the study of compulsion, that's where we didn't find the 130 Hertz as being efficient. And in that study, it's a PNAS paper published in 2021, led by Michael de Goulet. What Michael did, he was training rats to first escalate their drug intake. They had their DBS electrodes implanted during the process of escalation so that he could record their local field potential through the stimulating electrodes during all the steps of the escalation. And while the animal were losing the control, he trained them in another procedure in which, if they want to press a lever to get access to a lever delivering cocaine, they take the risk to have 50% chance to get access to the drug indeed, or to receive an electric foot shock. And most of the time, they don't like the electric foot shock, so they stop seeking for the drug. Mm-hmm. But we have rats that are resistant to this punishment. So we consider they are the real addicted rats because they have first escalated their drug intake, so they've lost the control over their consumption, and they really seek for the drug, whatever the risk to receive an electric foot shock. And these animals, there are no way so far to discriminate them from the others, not given on the level of drugs they take during the escalation. We have no way to predict. But we thought, we have recorded their STN activity during the escalation of drug intake. So maybe they have a different kind of activity in their STN during the loss of control. And that's what actually we found. Oh. And it was not a beta activity for a change, but it was a very low frequency band, around 8 Hertz. So that was a theta, delta, I don't know, but some people, depending on where you set the threshold of the band, you can call it. We considered it was a theta band. So then we thought, that can be a predictor of vulnerability for addiction. And 8 Hertz. So if you have an abnormal activity, not at 30 or 20 to 2030 like we have in Parkinsonism, 130 Hertz reduces the abnormal beta. Can we correct also an abnormal theta with 130 Hertz? So first we had to prove that the 8 Hertz was actually the marker of vulnerability. So we took animals that were not resistant to the punishment. We stimulated them with 8 Hertz to mimic a fake abnormal theta activity in their STN during a re-escalation process. And then we retested them. And while before they were extinguishing their behavior, they didn't want to take electricity in their paw. Now they became all resistant to punishment.
Really?
So that's how we published. It's frequency dependent because 8 Hertz in STN can induce some compulsivity. And surprisingly, indeed, the 130 Hertz did not correct this behavior but it's a 30 hertz stimulation that was able to correct an 8 hertz. So maybe there is a shift. So you can correct an abnormal beta by 130 hertz, but if it's an abnormal lower frequency, maybe you don't need to go to 130 hertz to have a beneficial effect, but an intermediate frequency. So then it's very interesting consideration, because if you think that OCD patients have actually abnormal beta, but also abnormal theta, maybe it's not only 130 hertz they should receive to have a total recovery, or an optimal recovery, because it seems to be quite beneficial, the STN target for OCD. But
in addition, 30, you could do both actually with the new devices. They allow dual frequency. Yeah.
Or maybe we need to have a closed loop system. When you detect some theta, you would apply a 30 hertz, and when you detect some beta, you start an 130 hertz stimulation. That might be a most optimal way to do that. And that's what I was actually suggesting in the conclusion of our paper on compulsivity in rats, is that if we want to use STN DBS as a treatment for addiction, maybe it's not a simple picture as applying an 130 hertz stimulation, but we should be aware that maybe depending on what phase they are, if they are craving for the drug, or if they are in a moment where they have another type of problem, we should record the activity of their STN before sending the stimulation to know what would be best appropriate.
Interesting. Love that train of thought. Amazing. So, okay. So yeah, lots to think about here. Great perspective on closed loop. I think summarizing again very broadly the findings on addiction, you concluded, I think 10 years ago, that STN-DBS should be favored for treatment of addiction. You still think so, I think, in your talks.
Even more,
even more. So
I have now even more evidence than 13 years ago.
STN for everything. So, but it was 10
not
years ago already.
Everything.
No. But no. Sorry, no. Don't get me wrong. No, I love the STN, and we just have a preprint out where it's effective for Parkinson's, Tourette's, dystonia, and OCD. Different circuits, but it is an amazing structure. So I that was not meant as criticism. That was actually in favor of it.
It's an interesting target because it's rational. Because now we have clear evidence. It's data-based evidence that the decision to choose a target for addiction at the moment should be STN. And I really, every time I attend functional surgery meetings and I hear these surgeons and they say, oh, we go to accumbens for addiction. Why would you want to go to accumbens? Because it's a center of motivation. It's not a good reason enough.
Somehow accumbens,
in the contrary,
I don't know,
because
you're right.
if you know your textbooks, you should know that if you go at the core of the motivation system that is not known to discriminate different forms of motivation, you're going to reduce motivation for the drug, but motivation for everything. So it's totally irrational. And we have data, and people just keep ignoring them, and they want to stick their electrodes in accumbens of patients.
Yeah,
go for it, but it's a total mistake.
I hear you, Christelle. I do. So when I wrote up the questions, the first thing I wanted to type was, why did it take so long to translate this into humans, or why didn't it translate into humans yet? Now,
very good question.
Told me that there was just right now out a case report of a first patient with mixed results. Do you want to talk about that? I think, first of all, why was it so complicated to get to that point?
It was really, really difficult. So honestly, I have to give credit to Luc Mallet, who is a psychiatrist from Paris, Salpêtrière, worked with Evagide. Again, you see, we always go back to the classics. And he actually read my rat paper, and immediately after the Nature Neuroscience 2005, he already wanted to do it in patients, in addicts.
15 years
So
ago.
he was all excited. So I said, "Ooh, hold on a second. Don't jump too fast." Even myself, I was not really convinced that we should go that fast. So I said, it's a lesion study. You're not going to do lesion in addicts. So let's wait for me to do it with DBS first in rats. Then we published in 2010 the PNAS paper with the DBS in rats for the same measures, motivation for food versus cocaine. And then he was still excited and he said, oh, now I write a PHRC. So that's a procedure you have in France to be allowed to do a clinical trial. So he actually obtained it quite quickly, maybe relatively. I don't know. I don't remember when he obtained it. But in the meanwhile, it was impossible. It was difficult to find first people who would accept to recruit patients for this indication, because it was not based on strong data, only my experiments in rats. So you want to believe in only 1 or 2 studies, really difficult. So, and then, so he found a colleague of us, an addictologist, Florence Vorspan. So she accepted in Paris to try to recruit patients. The problem is addiction is a really difficult disease.
Yeah.
By the way, it is a disease. Some people are still ignoring that. And then, so if you want to recruit volunteers for PD, OCD, it's easy because now you have nice videos you can show to the patients, and they all want to have the same thing. Oh, yes, it works. I want that.
Yeah.
I want that.
Yeah.
For addiction, there's nothing. There was a poor video from Damien Denis' group in Amsterdam for opiates indication, and they had implanted, not in the accumbens, but in the anterior arm of the capsule. But it's not apparently convincing enough for the patients to volunteer to be subjected to that heavy surgery. So it's a major point. Some people think it's only a matter of will, and then you can treat addiction just helping them to be more, to be stronger and have more will, and then they will get out of it. So no need to stick electrodes in their brains. And sadly, there are too many people still thinking like that. So it's a really difficult issue. So we had first the difficulty at finding even the people involved in the project, and then after to recruit patients. And this case is actually not an optimal case. So to cut the story short, it's really easy. Now the guy is perfectly fine. He has no stimulation anymore. He takes no drugs. He's perfectly fine. But we can't say whether or not the surgery did anything to him because the guy had too many problems initially. So he was really, really coming from a background. He was left on his own, living almost in the street. He had an agoraphobia that Florence treated while he was in the service. For 2 years, he's been nursed by a whole team. He had a flat for himself. He had all fantastic conditions. So it's so much all intermingled. What can you draw as a conclusion? You can't say much. So the guy is perfectly fine, but maybe he would have been perfectly fine if we did a placebo surgery and just giving all the care he received for once in his life. And so, I mean, I shouldn't speak like this because it's not my hypothesis, but
sure. Yeah, no, but it's, I mean, it shows that you take it seriously. So it's a success story, right? You could also say, wow, he's perfectly fine now.
But we didn't write the papers this way.
Exactly. I remember it very differently. So I think it was, screening over it, it read that it really didn't work so well.
No. She was really on a pessimistic aspect, she wrote the paper. So saying they couldn't see any change in the craving, but one thing that is not written in the manuscript, and I told her many times that I wanted her to refer to it, but maybe she thought it was too much anecdotal to be mentioned in the paper. But when they started to set the parameters of the stimulation, while they were increasing progressively the intensity of the current, at some point, the guy said, "Oh, I feel so well. I've never felt that well for so many years." They didn't stop at that stage because the procedures they had validated, with the law, with the thing, you have to apply what you've said you would do. So Luc had written the procedure as to apply a similar stimulation he applies to OCD patients.
Okay.
So he went further to reach the parameters for the OCD patients. Maybe this guy didn't need that much. And it would have been much more optimal if we were where he said he was feeling so well. And that's one of my regrets. I would have liked it to be stimulated at the parameters he said he enjoyed it. Because in our rats, we showed, it's a publication in preparation, they can self-administer the deep brain stimulation of STN. So it is rewarding by itself. And we can replicate that with optogenetic inhibition of STN neurons. They can press for it. So it had an effect on him.
Yeah.
Transient. And then I would have liked it to be pursued better, but it's really difficult. So I really hope someone is going to try again.
Are there more patients
volunteers? Sorry?
Are there more patients planned and just not been written up in this trial, or was it a single shot trial?
No, they were 3 or 4 planned. They haven't been included yet.
Okay.
And there may be in the future, but I'm not sure. And I really wish there would be. I know that Bomin Soon Service in Shanghai was planning to do it. Because they were planning to implant 60 accumbens. And then after listening to my talks, they said, oh, maybe we should try STN for 30 accumbens and 30 STN. Then I went there and I got a bit scared and I was like, oh, but of course, there might be side effects. And especially with all what we're discovering at the moment regarding emotions, that's maybe it's important to monitor well these patients, but the side effect may not be that terrible and maybe not less terrible than accumbens stimulation. I don't know what they're going to see with accumbens stimulation, but we'll see.
One thing I remember when talking to Valerie Woon is that it's a criminal offense to be addicted in China, right? So that makes the whole situation quite difficult to recruit, and I don't even know how they do that because they need ethical votes.
Anyway, they had a difficult discussion with the authorities. So the first time they are caught, they are not, they are registered, but not imprisoned or anything. But if they are caught a second time, then they risk to be put in a psychiatric hospital where you don't want to go. And then, so that's what they were trying to deal with the authorities in Shanghai to be allowed to do that study, mean that they have to be a bit more flexible with the patients. So we'll see how it goes.
Okay, got it. Interesting. So your comrade Marwan has a really great paper on serendipity and discovery of human serendipity discovery. So we find, for example, that a lesion here leads to cessation of tremor. Then we can essentially mimic it. Like if there's a stroke, I think James Parkinson's, one of his patients had a stroke that got rid of tremor. After that, people try to replicate that for a while, right? Bussy and so on, even lesioned M1 and the internal capsule and so on. So And if you look at, I think Marwan essentially analyzes the field of and looks as how many wins do we have, like how many treatments really came from serendipity and how many really came from animal research. You could even play the devil's advocate and say that the monkey model, MPTP monkey model, where then Haggai had the SDN lesion and that led to SDN DBS, came from frozen addicts in humans, right? So the idea, again, in a way, came from serendipity in humans, and then we went back to the animal. So I'm playing devil's advocate here, but where do you see the role in animal research in DBS? And then also, given the difficulties you had, it makes perfect sense in the rat. Now translating it to humans so far, at least, is mixed. What's your take on this as an animal researcher?
Because the story of the DBS is actually really coming from animal work that was highlighting the abnormal activity of STN and all this. It was the fashion, it was the beginning of my PhD, so I recall well that there was this passion in the basal ganglia community to consider this imbalance between and functional antagonism between dopamine and glutamate. So it comes from the late 80s.
Wait, but we had pallidotomies before that, right? And they worked great. And then I think Benabid even wanted to go to pallidal DBS first, right? So I think we have to be careful here.
The reason why STN was a target of interest is because it was the only glutamatergic system within the basal ganglia. Yeah. And that was shown to be hyperactive in case of dopamine depletion because there was this antagonism. So they tried the antagonist of NMDA receptor as a treatment for Parkinsonism with so many side effects. And then it was all this big fashion on that. And so, I mean, I really think that the animal work has been critical in that. And not only Haggai works was lesion of STN, showing the target is interesting. The technique of DBS was found by Benabid in the VIM of the thalamus. But then application of DBS in STN was first done in the monkey in Bordeaux by Amit Benazouz. Everyone always skips this step. And it really annoys me because it was applied there and it's not my group. So I really always advertise for them. I have no problem with that. And I'm happy to do it because it's unfair that they are not recognized. So the application of DBS in STN in monkey is the first work, Amit Benazouz 93. And then he moved to Benabid and that's right in the patient. So the animal validated the target before it was done in human. STN was not a target for PD before. So
I mean, not deliberately, right? If you look at the lesions Hassler made back in the day, they always try to avoid the STN for risk of hemibalism, of course. So they did not lesion it, but they did. If you look at the images, they had often subthalamic lesions that had great success and
but they would have never interpreted their result at the time.
You're right.
They were ashamed. Shit, we touched the corpus.
Yes. You totally agree.
So they would hide it. So you don't dare sometimes. I mean, if you work properly and you think rational, what do we know about this structure? You don't destroy M1 to help a Parkinsonian patient. So if one has an infarct and suddenly recovers the function, you're not going to lesion everyone in such a critical place in the brain.
Yeah.
So
I mean, they did in the time, but that was not a good idea. I totally agree.
No, exactly. Yeah. That was not a good idea because good science needs to be based on facts and facts that are replicated, studied with tools that you cannot always apply to human brain. So
I think I mean, I see 100% no doubt a lot of value in animal research. And I think I told you the same with the primate research. I think Marwan probably does too. It's more like it's still an interesting way, because in a way you said we kind of shape history towards the more human side, but you could also argue that our whole, publishing and funding machinery really favors animal research. All the Nature papers are mainly animal research, right? And the grants, bigger grants, are given to animal labs and so on.
But
So
Nature papers sometimes don't get cited because they are not that great. And
sure, but people
papers,
would want to publish there.
You have a much higher impact factor in the clinical journals. There are many clinical journals that have such a high impact factor, and so I don't know. It's
still prestigious, right? So everybody wants to, people want to publish in Nature, right? And then it's very hard to get in with clinical work.
Anyway.
We should. I know. That's a whole different discussion. I'm just saying
it's an old debate.
Yeah, I guess the whole theater is kind of. We put a lot of focus on animal research, and I think it's good, right? I'm not even challenging that. I just think it's an interesting point to see, okay, but if a lot of wins come from human observations that we could then refine in animals, right? That could be the other model where we say, hey, a lot of animal researchers study things in animals with optogenetics that we don't even know treats anything in the human, but we know a few things really work in humans. Better understanding them could be really cool animal work. But it sometimes feels like people are not so interested in that, like finding out better why DBS works or lesions work and so on. Anyways, I think there's certainly no animal is bad or good. It's all useful, and I'm a very big fan of basic science. I just thought it's an interesting discussion to be had with you.
It's just that I think this specificity we gained with these new technologies, allowing to target selectively a specific pathway, a specific subpopulation of neurons, we certainly can't have that in the clinical research nowadays.
Yeah.
So whether or not it has a direct translational impact, maybe not immediately. So it may take some time before we realize. And of course, also because, as I said earlier on, I'm not impressed by the type of question people ask with these new tools. So now they have specific tools. We are sure that they are recording dopamine neurons. While before you had to have to be called Wolfram Schultz or Tony Grace so that people would believe you were recording dopamine neurons. If you were a nobody, everyone was always giving you a hard time. Are you really sure the neurons you record are dopaminergic? Oh, yes, they have the waveform, blah, blah, blah. But so now they have the specificity, but do they show something so much different? And do they ask different questions? Oh, yeah, they rediscover movement in dopamine. Fantastic. But so the thing is, we have with animals fantastic tools nowadays. Whether we are using them properly is another debate. But I think they may have translational impact. But it may take some time. So, because we still can't do some of the things we do in animals in our patients. And that's good like that, I think.
Makes sense. I want to be mindful of your time. I've been taking a lot of your time. So I want to wrap up with some like rapid fire quick questions. Did you ever have a eureka moment in your scientific career where you said, wow, now I understand it?
I wish I would. No, but I get excited by difficult results I cannot explain. It's more exciting to me. If you have an hypothesis, you make an experiment and you find the results you thought it would be, it's boring. Then if it doesn't fit with any of the diagram you've studied and all of the textbooks you've read, then it is really challenging, and that's what I like. So no huge eureka moment.
Speaking about more the opposite, failures to also communicate that, did you ever think this was a big waste of my time? This didn't go well?
No, I never think we waste time. What we waste is the fact that we don't share the negative results enough. And we could save some time of other people somewhere else in the world. So that's really something we need to work on. But again, it goes with what we were saying about the big journals, etc. People want to publish, and it's so annoying. So I hope the archive system is going to save our system, and that we are going to get rid of these big editors and rely on our expertise to judge whether or not we should cite this paper because we read it on bioRxiv. It's really cool. They've done the job properly. Spend some time doing this instead of, because it's published in Science or Nature, it's good. No. Some work has not been replicated at all, and we all know that. So we should be really more invested in alternative systems. And so negative results is important to communicate. And no, you don't waste time if you have negative results, because you know something that your hypothesis was wrong, or there's something wrong in the system.
Advice for young researchers entering neuroscience in academia.
Sorry?
Do you have any advice for young researchers entering the field?
They've changed. My advices have changed over time. When I was young in my career, I was witness of the fact that it was difficult, of course, but not impossible. And every person, so that's what I was telling the young student coming to discuss with me. I was always telling them, if you're motivated enough, you'll find a job in academia or in private industry, whatever. But if you really want it, you can stay in research. Nowadays, I can't say that. And I see so many bright students who have to change career. It's heartbreaking. It's terrible. We've invested so much with them, and they are really good. And at some point, they just feel a lack of recognition. It's too difficult and too much pressure. And then they quit.
Is that why? Because we're training too many, or because it's harder to get grants these days than it was?
There is little money invested in research in most countries nowadays. That's a problem. So they want us to be to perform with little. So we are heroes, daily heroes, and that's what I keep telling all the people around me. We shouldn't feel ashamed because we are not competitive with some other countries. We do with what we have. So France is cutting the budget for research all the time. They have no respect for our work. And so doing what we do is already really good. It's a performance. And I see that in many countries, actually only a few countries currently are really investing in development of research. China, I don't know where else. I mean,
US Biden has increased the budget, I think, quite drastically.
In the US? Okay, good. But it's not the case in most countries. So it's a big problem. Then I think we still should train as many people as possible, because even if they go and do something else, which is heartbreaking, as I said, the training, the working with integrity, developing your curiosity, all things we were not able at my time to realize the richness of such a training and how it brings abilities you can use for any other type of job. At my time, we were trained like there's nothing else but academia. So if you don't do that, what are you going to do with your life? Nowadays, the young are not trained like that at all. So they think of alternatives. which is good. It's sad for us because some of them should stay with us, but at least now they know how to value the training they received, so which is good. So I think, no, we should not limit the training, but we should push our politics to invest more on us.
Sounds good. Advice for women in the field?
There's a long way to go. It's still tough.
I agree.
It's still really, really difficult for women in science in general. There are initiatives for diversity, equity, but there are still so many stereotypes, unconscious stereotypes, and that's the problem. So the first work we have to do is to make people realize, even though they would like not to be biased, they are. And we all are. And sometimes women are even tougher with other women.
Right, yeah.
So it's a really difficult thing. And when you sit in a committee, for example, and very quickly we ask you to give names of people to give a talk on a topic. Spontaneously, it's always men's name that come out first. Always. And then you have to correct to say, oh, it's a bit biased. And then suddenly, so who could we think of inviting? It's shocking. But even me, I have the same bias. And I have to force to say, oh, okay, I should be careful because I know there is a bias. So since I know, I try to anticipate, but it takes more time. And so there's a lot to do. But I hope that the next generation will benefit from all these initiatives. ALBA and EBPS, we have launched a group to work on diversity, equity, and things like that. So I see it's progressing.
Yeah, I'm also for this podcast, I started with the strong aim to have at least 50-50, but so far failed, unfortunately. And it's really because maybe it's also, as you say, biased. Absolutely, it's harder to think of like women sometimes, and I also feel like since neurosurgeons are still very male dominated, it's really a bit, especially under them, a bit harder to find people. So the other thing is that I realized, which again goes into the same direction, but I have really gotten more rejections from women that I ask. Probably just because men like to talk about themselves more, same kind of bias that they don't mind about, being on the stage. That is interesting too, that I feel like
I think it's, as always, I mean, even when I was in politics, it was the same thing, or giving responsibilities to a woman is always difficult because immediately the first question they will ask, am I qualified to do that?
Yeah.
Men never ask
Never
that
ask.
question.
Yeah.
They consider that if you ask them, you consider they are qualified. So they never question it. A woman systematically will start like that.
Yeah.
Do I think I am qualified enough to do that? And then they have the imposter syndrome.
Yeah.
It's a constant. And then after, can I afford to take some time to do that?
Yeah. True. Yeah. So in that vein, last few questions. So what do you think the future of the field will be of deep brain stimulation, neuromodulation? Is optogenetics coming for the humans? What are we changing? What are the next steps? What do you think?
Oh, I think it still has a nice future because there are still some groups are still working on indications, new indications. So refinement of the current applications are the quality of the electrodes, the way you can shape the current, etc. It's all a field that is still under development at the moment. So I think it still has. I don't think I will see the end of DBS while I will be still active in research and yet in photography. And I hope for new indications, of course. What optogenetic to human being could bring different will be based on DBS effect anyway. So they will still need DBS to develop opto further. And we are not there yet. Because when we see the difficulty to transfer the technology from rodents to primates, to have a specificity, a good expression of the virus in the human brain, make sure that it's all safe. Get all the official authorization. I'll be retired. It won't have started.
I agree. Any missed opportunities of the field, things we should be doing but are not? We heard more funding, but other things we should be doing.
Put STN in the reward circuit.
Yes.
Part of that, boycott the big journals and put your stuff in open access archive for free. Don't pay to publish. We pay already enough to do our own experiments. So we have to stop that. We need a revolution regarding the attitude with the big editors. And that's an editor speaking.
Where do you edit?
I'm how do they call that? Chief editor, maybe for psychopharmacology.
Oh, wow. Great. All right. So yeah, thanks for that. Any topic you would have wanted to discuss? I know we covered a lot of ground, but anything that I missed before we stop?
No, I think we talked a lot.
I know. Yeah. Thank you so much. This was a long conversation. So thank you, Christelle, one more time.
It was a great pleasure. Thanks a lot. And thanks to all my friends for their very interesting question. Marwan, Hagai.
Great.
That was great. And Garance.
Garance, yes. Absolutely.
Garance Meyer. She needs to be highlighted.
Absolutely. Thank you so much.
Thanks a lot. Bye-bye.